Okay, let's go ahead and start. We're going to continue looking at the chemistry chapter. We're going to take a look at the lipids today. We're going to start with that. Lab this afternoon is going to be, we're going to take a look at the bones of the skull. And that is not going to be on your first lab test, which you'll have next week. So we'll be looking at all the bones of the skull, and I'll try to make it as interesting as I can. Okay. Anyone have any questions before I get started? Okay. Having said that, I'm going to go ahead and open up the slide on chemistry and see where that gets us. Okay. So we had wrapped up last week on Wednesday. We had wrapped up the section on carbohydrates. Carbohydrates, of course, are carbons and hydrogens and oxygen in a 1:2:1 ratio, and with usually 1 carbon, 2 hydrogens, and 1 oxygen as the repeating sequence. In here, the carbohydrate that we use the most in our bodies is glucose, which has C6H12O6, 6 carbons, 12 hydrogens, 6 oxygens. So now we'll move on to the lipids, the fats. We call them the triglycerides, for example. These are the neutral fats. If it's solid at room temperature, it is a fat. If it's liquid at room temperature, it's an oil. So animal fats, butter, lard, fat trimmed off of meat is all it's an animal fat. It's solid at room temperature. The oils like corn oil and canola oil and olive oil are all liquid at room temperature. And they are also a fat. They are the plant fats. Every type of fat, when I say, how about I say triglyceride? Every type of triglyceride is made up of 3 chains of fatty acids attached to a glycerol molecule. And what that looks like is a structure something like this. The glycerol has 3 carbons here. It's an alcohol, so it has an alcohol group on the end. Anytime we have the OL ending on a word, it's an alcohol, like glycerol. This is glycerol, so that makes it an alcohol. And we have a fatty acid chain attached to each of the carbons. These are the fatty acid chains. And all they are carbons and hydrogens and carbons and hydrogens and carbons and hydrogens, usually with an oxygen or an acid group at the end. Acid group. Okay, group. Anyway, acid group. So this is what a triglyceride is. It's 3 fatty acid chains attached to a glycerol alcohol, and it forms solid fat, it forms liquid fat. We use fats in our bodies for storage of energy. In our muscle cells, we store lots of fat inside the adipose tissue so that we can draw on that fat for energy. There is more energy in a gram of fat than there is in a gram of carbohydrates. A gram of fat contains 10 calories of energy. A gram of carbohydrates only contains 4. We also use the fat, the triglycerides in our bodies for insulation. Underneath our epidermis, we have the dermal layer of living cells. And underneath that, we have the hypodermal layer where we have our fat deposits. And that's the fat there is designed, fat there is designed to keep us warm or keep us cool. It also functions as a layer of protection. Many of our organs in the viscera, in the abdominal cavity, have no protection other than a layer of fat that surrounds them. They don't have any bones to protect them, like the kidneys set up high in the back wall of our abdominal cavity. And the only thing that protects them besides the skin and the muscle on the outside of the abdominal wall is that they're embedded in a layer of fat. So energy storage, insulation, and protection are the big three things that we use our adipose tissue with our triglyceride fats inside. So now this is what a much better illustration of what a triglyceride looks like. You can see on the left-hand side, the glycerol, the 3-carbon alcohol, and these are the fatty acid chains, which are nothing more than long chains of carbons and hydrogens linked together, and they have an acid group at the at one end of the chain or another. In this case, this is what we refer to as the acid groups. Yeah, so it has a carbon with a double bonded oxygen and another oxygen hydrogen hydroxyl group attached to it. So this is what we call the acid group. See if I can spell it better this time. There's the acid group. That's what makes this chain a fatty acid. So we always have 3 fatty acid chains making up our triglyceride attached to the alcohol. And they're all identical in structure. They all have carbons and hydrogens. Now, what they represent also is because of all those covalent bonds, between the hydrogens and the carbons in here, we have a lot of potential energy. We've got a lot of covalent bonding taking place between the carbons and the hydrogens and the carbons and the carbons. And all that potential energy is available to us. That's why fats are a great source of stored energy in our bodies. They have more covalent bonding going on. Carbohydrates, we limit that, even when all the glucoses are linked together, we still have more covalent bonds in fats than we do in glucose or glycogen. So the potential energy is always greater in the fats simply because there are more covalent bonds. Okay. Now so this is what your fat looks like. 3-carbon glycerol attached to 3 fatty acid chains. Now we can refer to these triglycerides in one of 2 ways. We can call them a saturated fatty acid. You may have heard the term saturated fats. You hear it on TV, on advertising all the time about saturated fats in food, like in oils and butters and margarines and things like that. If you have a fatty acid and the fatty acid is saturated, what that means is that for every carbon atom in your chain, so here's your fatty acid chain right here, your acids down here at this end. You're going to have every available space held together with a single covalent bond, and every space for a hydrogen atom is going to be filled up with a hydrogen atom. So a saturated fat has its maximum number of hydrogen atoms available, the maximum number that it can possibly have. It'll be filled up, it'll be saturated. There are no gaps, there are no spaces. And a triglyceride that is made up of saturated fatty acids will always be solid at room temperature. Saturated fatty acids are solid at room temperature. Butter, lard, and then of course the animal fats are all solid at room temperature because the fatty acid chains are saturated. And we've all seen animal fat, we've all seen and eaten butter. You may or may not have eaten lard that you're not aware of. If you like to eat biscuits, you probably may have eaten biscuits made with lard, because lard is used as a shortening, just like Crisco is used as a shortening. Now, the fatty acid, this is a much better illustration here. There's a saturated fatty acid, similar to what I drew, but you know, see how the carbons are all linked together with covalent bonds, and all the carbons have hydrogens attached to it. So there are no empty spaces here. This is a saturated fatty acid. Now, if we have a saturated fatty acid, we got to have an unsaturated. And the unsaturated fatty acids have a double bond between two carbons. And that means that we don't have hydrogens available here and here, because the double bond is sharing more electrons. So this double bond here, there are no spaces for the hydrogens to bond here. So we don't have those extra hydrogens. We have this double bond between the carbons. So we're sharing, you know, the carbons are still happy because they're sharing more electrons than they were sharing on a single bond, but we don't have room for any more any hydrogens to be at that spot. That's an unsaturated fatty acid because we're missing some hydrogens. Now, the unsaturated fatty acids are going to bend at that bond. They're going to bend and kink at that double bond right here. By bending, they may even pivot and rotate around the double bond, you know, so that they not only are they twisted away, they may be circling in here and rotating around the bond. So they are not lined up in a straight line like the other fatty acids are. And that is significant, because if you have an unsaturated fatty acid, unsaturated fatty acids, because of that kinking shape, are liquid at room temperature, thanks to what we call van der Waals attraction. So depending on the length of the chain, it can be anywhere from up to 20 carbon atoms. A saturated fat will have every carbon attached to at least 2 hydrogens and maybe even 3. It will have the maximum number of hydrogen atoms attached to it. An unsaturated fat has at least 1 double bond. So if we look at a food label and it says unsaturated fats, you know, how many grams of that, or what the percentage is. An unsaturated fat it will have 1 fatty acid chain with a double bond in it. If you have a polyunsaturated fat, it will have multiple double bonds. So you could have a fatty acid that's polyunsaturated. You know, here we see there's our carbons and there's our hydrogens here. But now we have a double bond here. Now we'll have a hydrogen and a hydrogen. And we'll have a double bond down here. So now we have we can kink here and we can kink here. And so we'll have like a double bend in there. We call that a polyunsaturated fat. And there's a term we're probably all familiar with because polyunsaturated fats, you know, we hear a lot about that as far as how healthy a polyunsaturated fat is. Unsaturated fats are oils. Unsaturated fats are liquid at room temperature. Saturated fats are solid at room temperature. Now, from a health perspective, what does that mean? Well, we see saturated fatty acids held together by van der Waals attraction. Saturated fatty acids held together by van der Waals attraction. Remember, van der Waals is the weakest of all types of bonds. It is even weaker than a hydrogen bond, but a van der Waals attraction occurs when the electrons circling the atoms, circling the nucleus of the various atoms, will randomly, occasionally give a positive or a negative charge to the side of a molecule, to a long chain molecule like a fatty acid. Will occasionally change the charge on the side of the molecule from positive to negative or negative to positive. At the same time, the adjacent molecules, the adjacent fatty acid chains are doing the same thing. In other words, they're attracted to each other. Van der Waals attraction is a slight positive and negative attraction between these fatty acid chains that cause them to be solid at room temperature. They're attracted to each other. They don't twist, they don't bend, they don't kink around. They are attracted to each other magnetically, electrically, electrostatically. It's a very weak bond, and it breaks and forms and breaks and forms, but it breaks and forms enough so that we see it, we perceive it as a solid, like butter or animal fat or anything like that. Food, fats that are derived from animals are all saturated fats. They're all held together with van der Waals attraction, making them solid at room temperature. The unsaturated fatty acids have double bonds that causes them to kink and twist and bend at double bond. So there is no chance for van der Waals attraction to take place. They don't stick together. So they're liquid at room temperature. The oils, the plant oils, the olive oils, the corn oils are all going to be liquid fats because they all have double bonds in there. They're all unsaturated fats. No chance for van der Waals to occur. You can see, here's a saturated fatty acid here, and down to bottom, there's one next to it. It has a bend in it somewhere. And so it's going to pivot or twist or turn around the double bond. There is no van der Waals attraction occurring because there's too much of a gap, and it's twisting and moving all the time. They never get to line up. Okay. Unsaturated fatty acids are generally considered to be healthier for us. We say it's unsaturated because we don't have all the spaces for hydrogens to be there. So any of our plant oils are generally considered to be unsaturated. They are considered to be healthier for us than the saturated fats. And of course, but it still comes down to how much of this stuff do we eat? Do we eat a lot of saturated fats? Do we eat a lot of unsaturated fats? Is it possible to have health issues if we eat a lot of unsaturated fats? Yes. And just because it's healthier for us doesn't mean we can eat more of it and not have any issues. It still comes down to how much of it do we eat? How much do our patients eat? Now, let's see if this works today. And why aren't we?
All fats.
There we go. Let me start that over
fat
again.
Molecule, 3 fatty acids bind. Butter, lard, margarine, and salad oil are composed of lipids called fats. To make a fat molecule, 3 fatty acids bind to a molecule of glycerol. For this reason, fat molecules are technically called triglycerides. Fat molecules do not mix with water because they have 3 long hydrocarbon tails. Cells use fats for energy storage because the tails hold more potential energy than other biological molecules.
Okay, that's a nice little summary of what I've been saying here. Energy storage, these fatty acids, these liquid fats, these solid fats do not mix with water because of the long hydrocarbon chains here. And so if you add oil to water, it's going to float on top of the water, for example. So let's move on. Now, here are some unsaturated fatty acids. Here we see, on the very first one is called butyric acid. It's a saturated fat. It's a little fat, a little chain, but it's still a saturated fat. It has all of its spaces available for hydrogen, filled up with hydrogen. But here we see oleic acid here in the middle. It's a mono unsaturated fatty acid with one double bond. It is an oil. When that shows up in a triglyceride, that's going to be a liquid. And here we see linoleic acid, polyunsaturated, because it has two double bonds in there. And so it will kink in two locations and twist around those kinks. And so we see monounsaturated and polyunsaturated. Many food labels will show you the unsaturated, polyunsaturated, and the monounsaturated content, fat content of the food, that you're eating, depending on what it is. Okay. Now there is a process where those polyunsaturated fats can be broken and additional hydrogens added back into the chain. The term is hydrogenation. An unsaturated fat can be turned into a saturated fat by the process of hydrogenation. You can take corn oil and add hydrogens to it, break the double bonds and add hydrogen to it, and you can get a corn oil solid. And it will be, which is essentially what shows up in margarines. Margarines and shortening are usually based off of corn oil, for example, and they are hydrogenated. The hydrogen bonds, the covalent bonds between the carbons are broken, the double bonds are broken, leaving a single bond, and we add hydrogens to it, and now we change the whole nature of the fat. It goes from being a liquid to a solid, and we can use that as the basis of making shortening or making various types of margarines. The problem, of course, is that if you know what Crisco looks like, you open up the can of Crisco and it's white. It's just this white pasty stuff, okay? Well, that's what margarine looks like too. So we have to add flavoring agents to the margarine. We have to add coloring agents to the margarine. We have to add salt to it. We have to add salt to Crisco to give it any kind of flavor. So Crisco is used as a shortening in baking for pie crusts and biscuits and stuff like that. What shortening does is, as it breaks apart, it releases lots of water vapor and steam, which causes the pastry dough to form very thin and delicate layers of pastry. It makes a biscuit very light and fluffy. Without shortening, your biscuit has the texture of a hockey puck. Excuse me. So you would have very tough baked product, a pie crust. We like our pie crust to be very light and delicate. We don't like them to be hard and, eat the filling and throw the crust away because it's so painful to eat the crust, or it has no flavor. This is what we use shortening for. We use margarine in place of butter because margarine is supposed to be healthier for us than butter. Margarine is simply based off of corn oil that's been saturated, hydrogenated and saturated in, but we still have to add the flavoring agents and the coloring agents and salt to make it taste good for us. So we can hydrogenate corn oil to make margarine. We can hydrogenate peanut oil to make peanut butter smooth and creamy. Years ago when peanut butter was first marketed, you had to stir the peanut paste in with the peanut oil. And this was like, 100 years ago or something like that, whenever. At some point though, the food industry discovered that if you hydrogenate the peanut oil, you can make a very creamy peanut butter. Furthermore, hydrogenation improves the shelf life of the product. The margarine lasts longer. The Crisco lasts longer. The peanut butter has a much longer shelf life, and it's stable as far as food products go. The problem with hydrogenation is it creates a type of fat known as a trans fat, and trans fats are implicated, artificially made trans fats, as well as naturally occurring trans fats are implicated in increased risk of heart disease because of the ability to carry fats around our body. Now, naturally occurring trans fats, though, are usually showing up in dairy, from cows. Artificially produced trans fats are banned. If you look at a food label, you will see trans fats should always be 0 because of the higher risk of heart disease here. Now let's move on. These are all unsaturated fats. We've all seen, country crock and shed spread and, any of these other varieties here, even Land O'Lakes butter also has their own margarine, but to get it to taste good and to appear to look palatable, to presentable, all these ingredients have to be added into it. During World War II, margarine really hit its stride, but the dairy industry, as far as being produced, but the dairy industry said, no, you can't sell margarine as a yellow product that tastes like butter. You have to sell it like it is in its white appearance and add a flavoring agent to it. And so you can see in the advertising there that they had a color pack. You would take the margarine in the bag and add the dye packet to it with the flavoring ingredient and mix it, squeeze it up, and then you would have a bag of margarine. And somewhere after World War II, the food industry sort of overwhelmed the dairy industry. Now, and if you go into Food City or Walmart or any other grocery store, you would see that most of the spreads are margarines. And there's a very much smaller area just for the butters, so, but again, it comes down to what's the trade-off? You're trading chemicals and flavorings and salt for animal fat. And then it comes down to how much of that do you eat on a daily, weekly, monthly basis? How much of that will your patients be eating? There's nothing wrong with eating butter. There's nothing wrong with eating margarine as long as it's not done excessively. Because we can tell your patients that they can eat whatever they want as long as it's in moderation. Excessive consumption of anything is always going to cause some consequences. So now let's move on here. Trans fats, as I said, produced in meat products and milk products and in milk, naturally occurring. That's not regulated. But again, what do trans fats do? Trans fats are going to affect our cholesterol levels. In our body, you've heard of cholesterol. We know that cholesterol is an important fat. We'll talk about cholesterol here in a few minutes. But we also talk about the good cholesterol and the bad cholesterol. And that's not really the best term for it, but it'll do. The bad cholesterol is what we call low-density lipoprotein. And low-density lipoprotein will take fat and cholesterol, it will carry it from the liver, where it's generally formed. Cholesterol gets formed in the liver, all of our fat ends up in the liver. These low-density lipoprotein molecules will carry, lipo means fat, so it's a fatty protein, will carry fat and cholesterol out to the cells for the cells to use, because cells use fat all the time. Excess cholesterol and fat, once the cells take what they need, will be deposited in arteries, in the brain and in the heart. And as that accumulates, it's a leading cause of heart disease. So these trans fats are going to increase the level of these low density lipoproteins. So they're going to carry more excess fat and excess cholesterol to the vessels of the heart and the brain. And we see a buildup of fatty deposit then on the inside of the coronary arteries. That leads to heart disease. It leads to a heart attack. We may have a stroke in the brain as these vessels become blocked with these fatty deposits. The high density lipoproteins, on the other hand, the good cholesterol, they take fat and cholesterol away from the cells and bring it back to the liver for destruction. So we take the fat that we need and the excess is brought back to the liver for destruction. So we don't accumulate it around inside our arteries. The high density lipoproteins, the HDLs, the good cholesterol, are going to keep the amount of fat deposits in our arteries down, way down, keep our arteries open. It is the heart healthy type of cholesterol, the HDL, the high density lipoprotein, dragging along the excess fat to be broken apart in the liver. So, which is why we prefer to have a much higher number of HDLs than LDLs. We want more good cholesterol than bad cholesterol. We want to have less fatty deposits in our arteries and our veins. And the problem with trans fats is that it pushes the other direction. If trans fats get formed, or if we eat food that contain trans fats, we're going to get more of the so-called bad cholesterol, more LDLs. You know, a person that eats lots of dairy products and lots of meats to the exclusion of anything else, you know, lots of dairy, lots of meat, is more likely to be a candidate for heart disease than someone that doesn't. Someone that maintains a more balanced diet, someone who has, you know, more vegetables, more leafy greens, more carbohydrates than fats and you know, large amounts of protein like that. Nothing wrong with eating meat or beef or chicken or fish or egg or dairy. There's nothing wrong with eating any of that stuff as long as there is variety and moderation in what's eaten. Yeah. Okay. Something else that happens to us is if we eat excessive, as we also like to say here, conditions of excess nutrients. We eat more than we should. We eat more than we're burning up. The adipose tissue the adipose cells will bind onto the fatty acids and form triglycerides and store the fat inside the adipose tissue. The adipose cells are only a storage facility for fat. The term here is lipogenesis. We will create fat from excess fatty acids and glycerol. So a diet high in fat will often lead to significant weight gain as we accumulate the fatty acids and glycerol in the adipose cells. Lipogenesis, the creation of fat. When we do need that fat to break it down, to release energy, which we do all the time, you know, we don't always get all of our energy from glycogen. The process is called lipolysis, the rupturing, the lysing of fat cells. So now something else that I don't think I mentioned, but I will mention when we get into chapter 24, metabolism, is that a diet high in carbohydrates will also lead to fat buildup. Our bodies will take carbohydrates as glucose, let the glucose into the cells to power the cells. Excess glucose ends up in the liver as glycogen. The liver can only take so much glucose and can only make so much glycogen. The cells can only store so much glycogen. Excess carbohydrates get converted to fat and stored in adipose as fat tissue. A diet high in carbohydrates can also lead to accumulation of fatty deposits in the adipose cells. So you can't win. So that's what the triglycerides are doing for us. The triglycerides are necessary in our bodies for energy and for insulation and protection. If some is good, more is not necessarily better. Again, excessive levels of anything are going to cause us problems. That was the first category of the lipids. The second category are the phospholipids. The phospholipids are what we use to make up the membranes, the cell membrane of every cell, the outer covering of our cells. Phospholipids are similar to a triglyceride, only we kick off the third fatty acid chain and add a phosphate instead. And so it looks something like this. Here we have, we normally have the 3 carbon, carbon, carbon with the fatty acid chains. But what we do to get this phospholipid is we take the 3 carbons, 2 fatty acid chains, but then we attach a phosphate group here. So we end up with a molecule that looks different. It has a phosphate head and 2 fatty acid tails. It looks like this. And the cell membrane of all of our cells are made up of these phospholipids. It's a double layer where we have the outer layer that looks like this with the fatty acid tails. Why did I draw so many? And the inner layer looks like this. So the phosphate ends face the inside of the cell and the outside of the cell. And we have what we call a bilayer, a double layer of phospholipids. And this is what our cell membrane is made up. And this is the cell membrane structure. It looks like this. And that's what protects our cells, the inside of the cells, from everything. It's a very restrictive barrier that keeps many things out. It's very selective as to what comes in and what goes out. So anyway, these are phospholipids. This is probably the most important use for phospholipids in our bodies. Now, the third category, so we've seen triglycerides and we've seen phospholipids, are the steroids. Now, the steroids have a 4-ring structure you see down at the bottom. That's a steroid. And the most important steroids in our bodies are going to be cholesterol. So an individual that says, I'm never going to eat cholesterol because it's bad for me, that's not a good idea. Our bodies need cholesterol. Our bodies will make cholesterol if we don't eat it. We'll still maintain a certain level of cholesterol. Cholesterol and vitamin D and the sex hormones, estrogen and testosterone, and bile produced by our liver, stored in our gallbladder to break down fats, are all built off of steroids. We need cholesterol in our bodies for cell structures to reinforce the structures of the cell membranes. Vitamin D allows us to absorb calcium in our gut. The steroid hormones, the estrogen, testosterone, and the hormones from our adrenal gland are all derived from these steroids, as well as the bile salts that break down fats for us, also derived from steroids. So everything, we have some very important roles for the steroids, and they all look very similar. They all have a 4-ring structure like you see here, and the functional groups that are attached to the ring are all slightly different. The difference between estrogen and testosterone, female sex hormone versus male sex hormone, are just a few carbons and hydrogens. So, and the last category that we call technically other lipids, we have what are known as the icosanoids. The icosanoids are a type of localized hormone. Hormones are what we call chemical messengers that tell our cells to do things. The icosanoids work in very localized regions. The most important icosanoid is what's known as the prostaglandins. The prostaglandins help to control inflammation. You know, if we have a cut or a scratch, and it's, you know, we control how much is it going to become infected and inflamed, or is it going to heal? The prostaglandins play a role in that. The prostaglandins help to control our blood pressure. The prostaglandins are involved with uterine contractions. And so we have, but they're very localized effects. Now, these are a type of lipid that we use in special areas. It's not a, as we say, a body-wide effect. You know, blood pressure is going to affect certain vessels. Inflammation is only going to work at the area where there's an infection going on. So these prostaglandins have a very localized effect, as opposed to the systemic effect that a full-blown hormone would have. So those are the 4 categories then of our lipids. We have the triglycerides, we have the phospholipids, we have the steroids, and then we have the icosanoids as the 4th category. So that leads us to the 3rd type of organic molecules, the proteins. The proteins, they make up a large bulk of cell mass. We probably have more proteins than any other type of tissue in our body. It's sort of a toss-up, but all of our muscle cells are protein. For example, all of the collagen fibers in our bodies are protein. In fact, collagen is the most abundant protein we have. You know, it forms the tendons that attach muscles to bone. It wraps around our bones to make them strong. So it may be a toss-up there. But proteins do almost everything for our bodies. They are structural. They give us muscle cells. They can contract. They can stretch. They give us elastic fibers. So, and they have great strength. Chemically, proteins form enzymes. The ability, you know, the ability to control the rate of a chemical reaction for us, to make a chemical reaction go faster as a catalyst. And as, again, contractile. Muscle cells contract. We have contractile proteins that allow cytokinesis to occur. So we have multiple uses of proteins, both in a chemical and a structural approach. Typically contains carbon, hydrogen, oxygen, but we also add in nitrogen because proteins are made of amino acids. We will also occasionally have sulfur and phosphorus mixed in, depending on what amino acid we're using. And every protein is held together by what we call peptide bonds, because the amino acids that make up the proteins are linked with a peptide bond. One acid to the second acid to a third acid to a fourth acid, and so on. So we have these peptide bonds holding the protein together. Now, there are only 20 different amino acids. 20 different amino acids. Each amino acid held to another amino acid with a peptide bond. But we can have very large proteins made up of hundreds, maybe even thousands of amino acids. We have an almost unlimited supply of different types of proteins. I mean, when you consider the letters in the alphabet, for example, if you look at your typical dictionary, there's several 100,000 words in a dictionary, and there are only 26 letters in the English alphabet. And so you can make a wide variety of words out of those letters. The same thing applies to our 20 amino acids. We can make an almost infinite variety of proteins. Now, every protein is made up of these amino acids. There's only 20 of them. Every amino acid has the same type of structure. It has what is known as the amine group, which is the ammonia side. It has the acid side. So this is the amine. This is the acid. And then coming off of this is going to be what we call the functional group. We designate it by a letter R. So whatever type of amino acid it is going to be designated by that little functional group there. They're all going to have an ammonia group. That's nitrogen and hydrogen. They're all going to have an acid group. It's the little functional group, the R group, that determines what acid it's going to be. And this is what an amino acid looks like, better than my drawing. So here is the ammonia group, the amino group here, nitrogen and hydrogen. And over here is the carboxyl group or the acid group, a carbon with a double bond with an oxygen and a hydrogen over here. And here is the functional group right there. And this side chain functional group is what makes the amino acid this one different from the other 19. They're all going to be different. Amino acids are then held together by what we call a peptide bond. We often refer to proteins as polypeptides because it's a long chain of peptide bonds linked together. A long chain of these peptide bonds are a long chain of peptides or amino acids, whatever, So we often refer to a protein as a polypeptide because many peptide bonds linked together. So let's see what we got.
Proteins are folded into a complex globular shape. Each protein molecule consists of one or more chains of amino acid monomers. The amino acids are linked by peptide bonds, so a protein polymer is often called a polypeptide. Because they are so complicated, proteins are usually described in terms of 4 levels of structure.
Okay, this is what a protein looks like when it's first made in the cell. Those little purple beads there are the amino acids, and they're all linked together with peptide bonds one to another. And when we lay down the protein initially, we say its primary structure is just a long chain of amino acids linked together. Nothing's been done to it yet. Most proteins don't function in their primary shape, in their primary structure, because that primary structure is just a long chain. We usually have to shape the protein into a unique arrangement in here. So here we see, one after another. These are all different amino acids here linked together. There's obviously more than 20 here. Consider hemoglobin, the protein in our body that carries oxygen in our red blood cells. That's 360 amino acids in length. So obviously we're repeating lots of amino acids, using them over and over again to make a chain that long. So the primary structure of a protein is simply a straight chain. That's what we see when this, a straight chain of proteins, a straight chain of amino acids, a linear sequence. But they don't stay in that pattern very long. Usually they're going to get modified and twisted and turned into a unique shape just for that type of protein. Some proteins are spiral, some are in a sort of like a folded shape. Some are clustered into 4 parts. So the primary structure, straight lines. Next level is what we call the secondary structure. The secondary structure has been folded a little bit, and we can form what we call the alpha helix, a coil. The alpha helix, you know, if our primary sequence, it looks like this with an amino acid and an amino acid and an amino acid, and so on. That's our primary structure, each one held together with peptide bonds. The secondary structure can either be what we call the alpha helix, which looks very much like a spring. We find the alpha helix protein in our hair because our hair will flex and return to its original shape. We find the alpha helix protein in the elastic fibers in our skin because our skin can stretch and return to its original shape. The pleated sheet structure looks something like an accordion. You know, you have a sheet that is sort of like this, you know, where you have, it's not a spring, but it's like taking a piece of paper and folding it back and forth on itself. And it looks like what you find in an accordion. If anybody ever plays those things anymore. Anyway, but what a pleated sheet does is gives you great strength. You know, so the fibers in our skin that give us a great deal of strength are pleated sheets. We see pleated sheets in spider webs that have great resilience to you know, wind and rain, that survive outside with lots of environmental impacts, lots of wind, lots of rain, and they're still there. You know, some very large spiders, the orb spiders, those black and yellow ones that we see in the fall, produce these huge webs that takes a lot of effort for them to be destroyed by, you know, wind and rain and, you know, movement, stuff like that. So the pleated sheets are a very strong type of structure. The tertiary structure is usually made up of multiple variations of alpha and beta versions embedded there. You don't just have a protein with just the alpha helix or just the beta, the pleated sheet. You usually have a sequence of those structures within there, and that becomes the tertiary structure. And the quaternary structure is a very large protein, and it has 4 sides to it, and usually consists of several tertiaries linked together with it. Let me see if we make this a little clearer here now. Okay. Now, primary structure, straight line. I like my drawing better. There's your alpha helix. It's like a spring. We see the alpha helix, as I said, in elastin. It's springy. Our hair, like I said, our hair and the elastin fibers in our skin. It is the hydrogen bonding between the proteins, between the structure of the protein, that causes the protein to assume its shape. Hydrogen bonds will occur in that straight line and cause the protein to twist into its spiral shape, or cause it to assume a pleated shape. The tertiary structure is when you have this long chain of proteins, this long protein chain, and it's folding back in itself to give you the helix or the pleated sheet, and it continues to fold back over in itself, still held together, assuming that unique shape by hydrogen bonds. And then, of course, your quaternary structure is multiple tertiaries linked together. Quaternary has 4 segments to it. And the classic example that is always used for a quaternary structure is hemoglobin. Hemoglobin molecule has 4 tertiary structures embedded in it, and it can carry lots and lots of oxygen. Okay. Now, if we look at types of proteins, there are only 2 categories. They are either fibrous, like skeletal muscle or smooth muscle or cardiac muscle, or they are globular. A fibrous protein is very stable. Our muscles are made up of fibrous proteins. They're usually a tertiary or a quaternary structure. They give us strength and support. Keratin, the waterproofing material in our cells, in our fingernails, in our hair, on the surface of our skin, is a fibrous protein. Elastin is a fibrous protein. It lets us have the elastic resilience that our skin does to be able to stretch and contract. Our blood vessels, our arteries, have lots of elastin because every time there is a surge of blood through our arteries, the elastin allows the elastic fibers in the artery allows it to expand as a surge of blood comes through and then snap back. Collagen fibers that give us great strength. You know, a strand of collagen is stronger than a strand of steel of the same size. And of course, collagen, there is more collagen protein in our bodies than any other kind of protein. And so this gives us, great strength. We wrap our bones with collagen. We use collagen to attach muscles to bones. Collagen is so strong that it will snap the bone before the collagen fiber itself will tear. And then, of course, all of our contractile proteins in our muscle cells, that allow our skeletal muscles to contract, our smooth muscles to contract, our cardiac muscle to contract. These are all fibrous proteins. They don't change their shape. Even after death, they don't change their shape. Even if, consider eating a piece of beef or chicken or fish, the protein in the fish, the chicken, or the beef doesn't change its shape even after it's cooked. The fibers in beef are still there as fibers. There is no change to their shape. They can't contract anymore, but they haven't changed. Globular proteins are different though. Globular proteins are water soluble. They are round, like a little glob. That's why they call them globular. They are extremely sensitive to temperature changes and pH changes. They can be broken apart if the temperature goes up too high in the body or down too low. They can be broken apart if the pH changes. We call it being denatured. They can be destroyed. They are usually a tertiary or quaternary structure, but they also have very unique properties. Globular proteins have active sites on them where special chemical reactions will take place. Enzymes, those biological catalysts. Enzymes have active sites that only work for certain reactants, certain things that come together at that active site. And if the enzyme it gets too hot or too cold, the active site shuts down. If it gets too acidic or too basic, the active site shuts down, and then the enzyme can't function. Our antibodies that protect us from infection are globular proteins. Our hormones, if they are amino acid based, they are also globular proteins, and what we call molecular chaperones. Molecular chaperones are the structures that take a simple straight chain protein in its primary structure and twist it all around to its final form. And they kick it out, the chaperone twists it around and then kicks it out so it can be a quaternary protein or a tertiary with a bunch of alpha helix and pleated sheets in there. That's what the molecular chaperone does. So, yeah. The term denaturation, I'll just say denatured. What happens when a protein, this doesn't happen to fibrous proteins, it only happens to globular proteins. A globular protein becomes denatured when it unwinds and its active sites no longer function, because these globular proteins depend on their three-dimensional structure to work. It's like a key or a lock. You know, if you take a key and you bend it in half, it doesn't work anymore. It's broken. You take a lock and smash the lock, it doesn't work. If the shape changes, it can't do its job. Globular proteins, if they are affected by temperature or pH, become denatured and they change their shape and become non-functional. If they're an enzyme, their active site becomes broken down and can't be used. Now, we can see a mild type of denaturation. If we get a little too hot or a little too cold, and we will recover from that, but it can become permanent if we have an extreme in temperature or an extreme in pH. Consider an egg. The white of an egg is albumin. The white of the egg is albumin, and that is a globular protein. And if you cook the egg, the albumin will go from being transparent, it will turn white, and it will solidify. You can't reverse that. That egg has been denatured. The protein in the egg has been denatured, and you cannot reverse it back to its active state of its liquidy, gooey, clear state. Because when you open up an egg, you got the yolk and you got the albumin. Once we denature it, we can't go back. And it becomes a permanent change. And that can happen to the globular proteins in our bodies if we get too hot or too cold, if we get too acidic or too basic, if we go without oxygen too long. When our cells start to die, the enzymes and the globular proteins within the cells start to shut down. And if that condition persists, then they will be irreversibly denatured and can't function. Okay, so an enzyme. Enzymes have a big role to play in our bodies. Enzymes are biological catalysts. Remember, a catalyst is something that allows a chemical reaction to occur at a lower energy level. Catalysts, we don't have to spend as much energy if we use a catalyst, because in our bodies, we can't alter the concentration of reactants. It's always very small for a chemical reaction. We can't alter the concentration of reactants. We can't alter the body temperature. We're sort of stuck at 98.6 or 37 degrees Celsius. We can't alter temperature. We can't alter the concentration of the reactants, and we can't alter the size of the reactants. We're going to be dealing with small concentrations and with very small particles. But what an enzyme does is allows a chemical reaction to work faster by simply flooding the area of the reaction with enzymes. The enzymes can speed up a reaction without being affected themselves. They lower the energy that you need to have a reaction occur. For example, if we want a chemical reaction to take place, if we're mixing up baking soda and vinegar in a test tube, okay, so baking soda and acetic acid, or let's make it sodium bicarbonate and acetic acid. We mix it together, it foams, it bubbles, we release carbon dioxide, and everybody's happy. Because we had lots of both. We had lots of acetic acid, we had lots of sodium bicarbonate. However, what if we only had one molecule of one molecule of baking soda and one molecule of acetic acid in a very large beaker of water? What are the odds of a reaction taking place there? Yes, we have the reactants. We have baking soda and vinegar, and we have it in a very large beaker of water. When are they going to randomly run together with each other? We may wait for days for those two molecules to hit each other and react. What an enzyme does is we will flood the area. If we added an enzyme to our beaker of water, and the enzyme was designed to grab onto two molecules of bicarbonate and molecules of acetic acid, vinegar, and bring them together. the reaction would go so much faster. Imagine this. Imagine you're in a classroom. All the furniture has been removed. You're just in a big empty classroom. There's two of you. You're both blindfolded. And you are released from opposite sides of the room and told to wander around until you meet the other person and shake their hand. So you're blindfolded. You have earplugs in so you can't hear them moving. And you're just walking around trying to encounter the other person. And it's a standard size classroom. How long would it take you to find the other person, to randomly run into them and shake their hand? Who knows? But imagine now that we put 20 people in the classroom with you, and their role is to take you by the hand and lead you to the other person so that you can shake hands right away. And then they let go. And then another person comes up and grabs you and leads you to the other person, and you shake hands. They are functioning as enzymes. They make the reaction go faster. That's what enzymes do. They bring the reactants together faster than they would normally occur randomly. That's how enzymes function. We make enzymes because we can't do anything about body temperature. We can't do anything about concentration or particle size. So we just use the enzymes to over and over and over again bring the reactants together. And so every enzyme has a location on it called the active site, and the reactants are called the substrates. And the substrates will land on an active site and come together because we flood the site of a reaction with enzymes. And those enzymes are specific for certain substrates. They only work on certain, each enzyme is going to be unique for its particular substrate. So for example, in our mouth, there's an enzyme called amylase. Amylase breaks down starch and nothing else. And so when we eat a carbohydrate, the amylase, which is found in our saliva, takes the carbohydrate and binds it to the active site on the amylase. The amylase binds the starch to its active sites. And we start breaking the starch apart into glucose. Nothing else is affected by amylase. Amylase can't work on protein, amylase can't work on fats, and it will tear apart starches because only starch can land on the active site of the amylase. And the starch then is the substrate. So we start the process of digestion in our mouth for starches, for carbohydrates. And we can have millions of reactions per minute, as opposed to the random occasional molecule running into another molecule and having a chemical reaction. So now enzymes usually have two parts. They have the protein portion called the apoenzyme, and they have a cofactor or coenzyme, which is usually a vitamin. A coenzyme is not a little enzyme or an assistant enzyme. It's just an organic compound that helps the true enzyme function, the protein function. So we have the apoenzyme and the cofactor. And I can't remember vitamins. We'll talk more about vitamins when we get into metabolism. Enzymes are very, very specific. They have a very specific substrate, meaning the reactant. Amylase only works on starch. Oxidase only works with oxygen, for example. Dehydrogenase takes hydrogens away from certain organic compounds, dehydrogenase. Anytime we have the ending ASE attached to a word, that means it's an enzyme. Hydrolase, oxidase, catalase, amylase, breaking apart starches, for example. So they are all going to be unique and specific for certain types of compounds. You know, amylase can't work on protein. Amylase can't work on fat. Lipase can work on fat, but it can't work because lipase, lipid, you know, lipase can't work on starch. They're all going to be unique. Every enzyme is going to be different for its very unique substrate. And this, remember, the substrate is just the reactants. And what they do, and to reinforce this again, enzymes will lower the amount of energy needed to start the chemical reaction. If you have those two molecules, one of acetic acid and one of sodium bicarbonate, and they're in a big beaker or just sort of randomly moving around here, it may take forever for them to combine. If you heat the reaction up, they would move faster, but you've had to apply energy to it. In our bodies, we can't heat things up to make it work faster. So we simply lower the amount of energy necessary. The one thing we can do is flood an area with enzymes designed just for those substrates and bring them together. And once we bring them together, they will have the reaction, and then we let go of it. You know, in the case of baking soda and vinegar, the enzyme we would use, they combine, and they release carbon dioxide, and they release water, and they release salt, and they're done. And now we have our enzyme ready to do it again with the next molecule that it encounters. We didn't have to heat anything up. We didn't have to increase the concentration. All we did was add lots and lots of enzymes. So let's see what this does for us here.
And is a protein that changes the rate of a chemical reaction without itself being changed into a different molecule in the process. Enzymes promote and regulate virtually all chemical reactions in cells.
So we depend on enzymes for just about everything. And so, you know, these globular proteins, very, very sensitive to temperature and pH changes here. We need them to do pretty much to control all of our chemical reactions and all of our metabolic reactions in our bodies. And just this little, you know, this is a good summary of how it works. There's your enzyme. There's the active site over here. These are your substrates. Perhaps we're linking amino acids together to make another protein. They come together. They have the reaction. They form their bond. We release the amino acids, and there goes the enzyme ready to go do it again and again and again. The enzyme itself does not get destroyed when the reaction takes place. And the problem here we have with enzymes, so now if you watch the animation, you can see the active site is perfect for the substrates. But if the enzyme becomes damaged by heat or pH, the active sites change, it becomes denatured. The reactants, the substrates aren't changed, but the reactant, you know, once it's denatured, the substrates don't fit into the enzyme anymore. And so once the enzyme is denatured, it can go back, perhaps. Here we see a typical healthy enzyme. Here come the substrates. They come together. They have their reaction. We kick them out. They're good to go. But now the temperature has risen in the body and or has dropped. It works either way. And the active sites on the enzyme are altered because the protein has become denatured. It has broken down. Substrates haven't changed, but they will no longer fit on the active site. They don't work. And again, this can be from temperature changes, either too hot or too cold, or pH changes, you know, too acidic or too basic. Yeah. And this is a good place to stop. We'll finish this up on Wednesday with the nucleic acids, the DNAs, the RNAs, and we'll probably have time to start looking at the chapter 3 on cells too, because this is a short section here. And I'm going to get us out of here. And lab this afternoon, again, we'll be looking at the bones. We're going to start bones. We're looking at the bones of the skull. Bones are not on the lab test. Your lab test will open up next week, and we'll stay open for 48 hours. And in the meantime, next week, we'll probably continue looking at bones. So more on that on Wednesday. So anybody have any questions? First lecture exam is going to be as soon as, it'll probably be, we'll finish chapter 2 on Wednesday, and our first lecture exam will probably be the week after, not next week, because you'll have your lab test next week. It'll be the following week. So, well, actually, since your lab test is on Monday, I could open up the lecture test on Friday of that week. That would work. You know. So, and I'll probably leave it open for several days for you. So think about perhaps the end of next week, not this week, but the end of next week for your lecture test to open up. So we're a ways away yet from that. We got comments. Okay. Okay, good. Yeah, so don't worry about the lecture test yet. But as soon as I finish this on Wednesday, I'll give you a good date for the lecture test. So I'm trying hard not to let you have 2 exams due in the same week. So, okay, any other questions? Okay, then I'm going to get us out of here. I'll see you all on Wednesday. Oh, I'll see you in lab this afternoon.